All right, hello everyone, and welcome back for more biotechnology. So let's talk a little bit about scientific methodology in a research facility, and we'll look at some specific examples to kind of give you an idea of the sorts of things that you'll be doing. So we talked about this previously, but regardless of whether an experiment or some production process, regardless of whether that's being carried out at a biotech company or in an academic lab or a government lab or whatever, the scientific method is followed. So the scientific method in a nutshell is, number one, state your testable scientific question or problem. And the word testable there is very key. You don't want to pose a question that can't be answered or can't be tested, right? So whatever your question or problem is, it's likely going to be based on prior observation, whether it's experiments in the past have led you to this point, things that you read in the literature have led you to this point, or some idea that you've had, something is going to lead to you asking this question or posing this problem. You want to develop a testable hypothesis. Again, testable means that certain experimental evidence could not only lead to affirmation of your hypothesis, but it's important that your hypothesis can be disproven as well. In fact, most hypotheses will be disproven. It's just the way things go. So once you have your hypothesis, you want to plan a valid and well-controlled experiment. So you can't just go crazy and go into the lab thinking you're just going to throw beakers all over the place and come up with some sort of data that leads to a conclusion. You've got to make sure that you design your experiment well. So a good experiment should only allow one independent variable to change between control and experimental groups. So your independent variable is essentially the nature of what you're testing in a particular experiment. So for example, if you're testing say if you've got plants or flowers to grow and you're testing how much water does it take to get a plant to grow, then you're going to make sure that all of your plants or all of your flowers get exactly the same conditions across the board except for that independent variable, which would be the amount of water that they get. So everything else has to be exactly identical. So in addition, you want to have ideally both positive and negative controls. So these are going to make interpretation of the results much, much easier. So a positive control is a group that will predictably give affirmation of the hypothesis. A better way of saying that is that a positive control is something that is almost guaranteed to give a positive result. It's basically saying, if my hypothesis is correct, this is what my experimental group is going to look like. So the positive control is basically just something to tell you that this is what things are going to look like if the hypothesis goes one way. A negative control is going to be the opposite. It will be a group that lacks the independent variable, and it's basically going to be, if your hypothesis is incorrect, this is what it's going to look like. So ideally, you would want to have your positive and negative control. A negative control should always be possible, but sometimes a positive control, for a variety of reasons, just isn't possible to have. So you try to do the best that you can. And then number 4, once you have your experiment designed, you conduct the experiment, you collect and organize data, you record observations, and you do the experiment several times. It's never enough to do an experiment just once. You have to do an experiment several times to confirm that your results are reproducible. So there are all kinds of weird things that happen in science. They should have scientific explanations, but sometimes they seem like they do not. So you do not want to get too crazy about the results of an experiment that are done once and then are never able to be reproduced. It's probably some weird thing was happening that you just are not aware of. So once you have confirmed that your results are reproducible, you come to some conclusion. And you do this based not only on the data, but also on statistical analysis that will tell you whether the data between your 3 or 4, how many experiments you did, are statistically significant. And this is going to be where you decide whether your hypothesis is confirmed or not. The statistical analysis. Are things statistically significant? Is the hypothesis supported or is the hypothesis not supported? If the hypothesis is not supported, It's not the end of the world. Like I said, most hypotheses end up this way. It just means that you need a new hypothesis. So you basically start over, you come up with a new hypothesis to the same problem or same scientific question. You design new experiments, and you just keep plugging away until you come up with something that works. It really requires a lot of kind of stick-to-itiveness. Okay, so let's look at a very specific experiment that the book puts forth. So we've already talked about how most biotech projects are in some way going to involve the manipulation of genes. So a gene being a stretch of DNA that provides the instructions for making a protein. In this little simulated activity we're going to do, we're not going to be manipulating genes. We are going to be manipulating genes. Yeah, you heard me, right? Okay, so with that little bit of comedy out of the way, here's going to be our stated problem. So the idea is that we want to create a faded look on denim jeans, not the kind of jeans that we were just talking about, literally jeans that you wear. We want to create a faded look on denim jeans by washing them with bleach, but we don't know what concentration of bleach to use. Obviously, with something like bleach, you could imagine that you could kind of go overboard, right? So we are concerned that too high of a concentration of bleach will damage the fabric, but too low of a concentration will fail to give that sufficient faded look that we're looking for. Okay, so let's come up with a hypothesis. Here's the one that I came up with. An optimal concentration of bleach that will give a sufficient faded look while minimizing fabric damage is going to be somewhere between 0 and 100% bleach. Yeah, right, genius. How'd you come up with that one, right? So that is kind of obvious. So we want some bleach, but we just don't know how much. 0% is just pure water and 100% is pure bleach. So we want something in between. We can specifically hypothesize that fading will be less efficient as the concentration decreases. So that's going to be if we try to focus on minimizing fabric damage. But on the other hand, if the concentration of bleach increases, we are going to see increased fabric damage. So these are the things that we're trying to consider. How much fading are we seeing, and how much fabric damage do we have to deal with? So in order to test our hypothesis here, to actually determine what is the optimal concentration of bleach to use, we are going to create bleach solutions that range in concentration. And this is a technique called serial dilution, which we will be talking about later in the semester. So basically the way a serial dilution works is you start with a stock solution. In this case, we are dealing with 100% bleach. And then you basically just start making one-to-one mixtures with water or some other solvent, depending on what you want. So we start with full-strength bleach, so that's 100% bleach. We mix it one-to-one with water, so 1 part water, 1 part bleach. So that is going to give us 50% bleach. So 50% bleach, 50% water. Okay, we're going to do that again. We're going to take our 50% bleach, take 1 part of that and mix it with 1 part water, so that is going to cut the concentration in half again, down to 25%. And then we keep doing it again. We could go to 12.5%, 6.75%. We could keep doing this over and over and over again, or 6.25%, excuse me. So we basically keep making different concentrations of bleach, and then we do have one negative control in here, and that is 0% bleach. That's basically just pure water. So that is going to act as our negative control here. So often before the real experiment is done, now that we have our different concentrations of bleach, sometimes a little bit of further optimization may be necessary. You don't necessarily want to just launch right into your experiment. You maybe want to stack the deck a little bit more in your favor. So some example of things that may need to be optimized, how much denim is necessary to be able to interpret the results. So you don't necessarily want to just use a whole pair of denim jeans for every one of those concentrations. You would go through a lot of jeans that way. So maybe we could just cut out a little patch of denim, but you would need to determine, okay, how much is too much, how little is too little. The book seems to have decided that a 10 centimeter by 10 centimeter square of denim seems to be perfect. So that's the sort of thing that you might decide on based on some optimization. Another one might be, how long does the denim need to be exposed to bleach in order to see fading? So you could say take 50% bleach or something like that and put take several different dilutions of 50% bleach and put a patch of denim in each of them, and then maybe after a minute take one out, after another minute take one out, and figure out how long does the denim need to be exposed to the bleach to really see what we're looking for. So again, the book seems to have decided on 1 minute here, so that's another thing that could possibly be optimized. And then also, do we need a reliable system of quantifying fading and fabric damage? So again, the book came up with an idea here. This is the sort of thing that would be determined experimentally. They came up with a ranking system in which you kind of have this scale down here for ranking the fading process on a scale of 0 to 10, where 0 is no fading and 10 is complete fading. And then we also have a system of ranking fabric damage from 0 to 10 again. So it's that ranking system that is going to allow us to create our data sets here. So we're going to keep track simultaneously for each one of our bleach solutions, to what degree did we get fading and to what degree did we get fabric damage. So in addition to the protocol of the experiment, any observations, any data that are collected, we need to record those and organize them into a legal scientific notebook. So this is going to be one of the first things that you do this semester, is create your own notebook and basically get it all set up to record data and to put your observations into it. So the rule of thumb for writing up an experiment in your notebook is that the experiment should be recorded in such a way that anyone, and I mean anyone, could pick up your notebook and replicate the experiment exactly the same way you performed it. So basically, leave no detail out. No detail is too insignificant. You want to make sure that your experiment is reproducible, and in order to make sure it is reproducible, you want to make sure that someone could do the experiment literally exactly the same way you did it. So again, leave no detail out. So what you're going to be collecting from your experiment is called raw data. Raw data can be a little difficult to interpret, so this is going to be you recording your rankings of fabric damage and fading, but again, raw data can be a little difficult to interpret if you're just kind of looking with fresh eyes at someone's notebook. So that is why it is very helpful to organize raw data into tables, histograms. You can see a histogram down here in the bottom right, or other types of graph that visibly demonstrate the conclusion made from the data. So the raw data themselves are still preserved in the notebook as they initially were, which is good because mistakes can be made in generating graphs and histograms, so you want to make sure that you have the raw data to go back to. Some other examples of raw data might be pictures or Polaroids that you take of the denim. You want to make sure that you have that for posterity's sake, because you may decide that the ranking you did maybe wasn't appropriate. So if you have pictures, you can actually go back to one of these experiments that you did months ago and maybe re-rank them. So you don't necessarily have to do the whole experiment again if you're in the business of maybe re-ranking them based on a new ranking system. So go ahead and pause the video for a second and take a look at these graphs and let me know what conclusion you would come to based on these data and graphs. So take just a minute to look at these and come up with your own conclusion here. Okay, so if we're coming to a conclusion, we are basically bringing the experiment to a close. So if we look back on our original hypothesis, we can go ahead and read it again. It says an optimal concentration of bleach that will give a sufficient faded look while minimizing fabric damage. It's going to be somewhere between 0 and 100% bleach. That's why we made our different dilutions, but 100, 50, 25, 12.5, and 0. So, however, we can specifically hypothesize that the fading will be less efficient as the concentration decreases and fabric damage will worsen as the concentration increases. So if you look at the data in these tables and graphs, the tenets of that original hypothesis do seem to be backed up by the experimental evidence. So if you're looking at color removal, at 100% bleach, we get we get 10s across the board. So 100% bleach totally gives you that faded look. 0% almost gives you no fading, right? It's just pure water, so we wouldn't really expect to see any fading anyway. So as we go from 100 down to lower concentrations, we go 10, 8, 5, 3, 0, 10, 7, 4, 2, 1, 10, 8, 5, 2, 2. So again, as the concentration decreases, the fading gets less efficient, okay? That was one of the tenets of our initial hypothesis. Okay, let's look at the fabric damage then. So, as we thought, at 100% bleach, the fabric damage is as bad as it could possibly be. We see, again, 10s across the board. In pure water, we get almost no fabric damage, and then as you go from high to low concentration, 10, 5, 2, 3, 0, 10, 6, 3, 2, 0, 10, 5, 4, 2, 1. So there, the trend very strongly says that as the concentration of the bleach increases from bottom to top, the amount of fabric damage is going to get worse and worse. And that is the other tenet of our hypothesis, and the data back that up. So, a last few things to consider before we move on. Once again, are these results reproducible? You have to show that the results of your experiment can be repeated again and again and again. Usually somewhere between 3 and 5 times is kind of the standard operating procedure. So again, this is why it is so important to record your experiment in very nice detail in your notebook so that you can repeat the experiment exactly as it was the first time, and you can do each experiment exactly the way it was done before. And then statistical analysis, which is something we're not going to talk about right now, is going to help us determine statistical significance. For example, is the fabric damage we see between 50% and 25% really that different? So that's something that could help us to answer that question. Did experimental error play a role in the results and data collection? So think about some things that could lead to certain conclusions that maybe are just due to experimental error. So maybe you didn't make your dilutions correctly. Maybe what you thought was 50% bleach was actually 75% bleach because you didn't mix things together well. Maybe you left some fabric in the bleach for too long. Maybe you left it there for 5 minutes instead of 1 minute. So those are things to think about as well. And again, this is why it is very important to record all of your observations in your notebook, including any boo-boos that you make. And it's okay to make mistakes in the lab. Everyone makes them. You just have to be absolutely sure that when you make a mistake, you record it in the lab notebook so that when someone picks up your notebook and looks at your data, they can see your data and be like, oh, wow, we got some weird results here. And then they can go to your observations and see, oh, it's because whoever did this experiment made a little mistake. And then that'll kind of help to make sense of things. And then the last thing to think of once you're done with this experiment is, okay, what's the next experiment for us to perform if that's applicable? So scientists and biotech people are always having to think several steps ahead. So you always want to have an idea while you're performing an experiment, if I get this result or this result, what is going to be the next step after that? If you don't think about those things in advance, you're going to have to just kind of sit at your desk for a while trying to rack your brains coming up with what the next experiment is. Rather, if you think of those things ahead of time, you can minimize the amount of time lost lamenting over a bad experiment or patting yourself on the back for a good experiment. You can decide, okay, what comes next. Okay, so it's not always going to be the case, but usually once several experiments have been done, or actually quite a few experiments, publication of your results is something that most labs are going to shoot for, right? So publications will help labs either secure their patent and get all the money from that, or secure funding through grants if you're in an academic lab. So it is the results of many, many experiments performed over the course of a year or more likely several years that are going to help form this cohesive model, this story that actually makes for publishable material. So it turns out there are very few general interest journals. There's Nature, there's Science, there's Cell, but that's about it. Those are really the only kind of general interest journals that I can think of off the top of my head. Most journals are going to be kind of very specific or esoteric. So most likely you would submit your work to a journal whose interests are in line with your manuscript. So if you are submitting a new protocol for the production of insulin, you might want to submit that to the Journal of Endocrinology or the Journal of, I'm making up something here, but the Journal of Biotechnology, Biotechnological Techniques or something like that. I don't know if that's a real journal, but I'm just spitballing here. So once you submit your manuscript for peer review, you write up the manuscript, including an introduction, kind of background information, materials and methods. How did we do our experiments? This is what we did. This is what we were thinking. These are our data. These are our conclusions. The peer review process is usually going to involve around 3 to 4 well-respected scientists in your field scrutinizing your work for not only its legitimacy, but also the scientific rigor, how well your experiments were done and controlled, how relevant is the work, does the work really need to be published. So if they all agree that things look good, they will recommend to the journal's editor as to whether or not your work merits publication. This can be a very painful process, as I know from personal experience. I've had more manuscripts rejected than I have had accepted. It's just kind of reality. You kind of have to develop thick skin in that regard. So rejection usually means that your manuscript contains some flaws, minor or major, usually major if you're just getting outright rejected, and it means that you need to address those concerns and correct those flaws before it's going to be seriously considered by another journal. So if your manuscript gets rejected, you've got more experiments coming, so you got to tighten up your story, tighten up your work. So usually more experimental evidence is usually necessary. So this is a screenshot of the title page of one of my manuscripts that actually did make it through the peer review process, thank goodness. So this actually took a little over 2 years worth of work to actually get to publication. So I had to do the experiments, had to write the whole thing up, had to decide which journal to submit it to. So you can see we submitted it to a journal called Redox Biology, not exactly a general interest journal. That's kind of very specific. So we went through the peer review process. They suggested some new experiments. We did those, and then finally we got it published, which is a very good feeling, I think you can imagine. All right, so that is going to do it for this particular video on the scientific method in biotechnology. Join us next time, and we will start talking about careers in biotechnology.